the peripheral nervous system, the neural crest cells here and the epibranchial placodes will form the neurons of the peripheral nervous system, or I can say the neurons of the sensory ganglia and the autonomic ganglia. The peripheral nervous system, here it is, the central nervous system, this is the cerebrum, here are the two big thalami, midbrain, pons, medulla and the spinal cord. So this is the basic arrangement of the parts of the central nervous system, and there are some nerves, third, fourth, fifth, sixth, seventh, 8th, 9th, 10th, 11th and 12th. So these are the cerebral nerves forming the part of peripheral nervous system, and here are the 31 pairs of the spinal nerves living and entering in the spinal cord. So these are forming the peripheral nervous system. The cranial nerves are having the sensory fiber, and the all spinal nerves are also having the sensory ganglia, having the pseudo-unipolar cells. The peripheral process of peripheral neurite is bringing the information from periphery to the central nervous system, and the central process is conducting the impulse from the pseudo-unipolar cells of the posterior root ganglia to the central nervous system. So the peripheral nervous system is formed by the 43 pairs of the cerebrospinal nerves, 12 pairs of cranial nerves supplying the head and neck, and also supplying the viscera of the thorax and the gut, and 31 pairs of spinal nerves innervating the upper limbs, lower limbs and the body wall from root of neck up to tip of the great toe. So there are 12 pairs of cranial nerves and 31 pairs of the spinal nerves. So basic of the nervous system, basic structure of nervous system is having two types of cells. The electrically excitable cell is the neuron or the nervous cell, neuron, or also known as the nervous cell. The neuron or nervous cell is the basic functional, structural and the electrical unit of the nervous system, that is the central nervous system as well as the peripheral nervous system. So the neuron is the functional, morphological and electrical unit of the nervous system. A typical neuron, I will draw a typical neuron here. There are different types of neurons, but here I will talk about a typical multipolar Golgi type 1 neuron here. A typical nerve cell or neuron is having two parts. The first is the cell body or soma, also known as perikaryon. So this part is cell body, also known as soma, also known as perikaryon. The nervous system is built up of trillions of neurons. Trillions of neurons are having different structure and size and shape according to location and function. But a basic, or I can say the most basic neuron is multipolar neuron of Golgi type. First, the cell body or the perikaryon is having a well-defined large nucleus, because neurons are metabolically very active. They have to generate, they have to propagate, they have to integrate, they have to interpret the information received from the periphery, and they have to make decision. So metabolically very active cells, neurons having large well-defined nucleus and very well-defined large nucleolus. These cells are having Golgi apparatus near the nucleus for modification and packaging of the proteins, and they are having the unique feature or unique structure. These are the rough endoplasmic reticulum known as nasal substance or nasal bodies. So these are having groups of ribosomes and rough endoplasmic reticulum. So these are the Golgi apparatus. These are having the nasal substance or nasal bodies, having nucleus and large nucleolus. Besides these subcellular organelles, these are also having lysosomes. These lysosomes digest the foreign particles in the cell. So in old age, where the neurons are degenerating, these lysosomes are forming the lipofuscin pigments. Lipofuscin. It is also known aging pigment, or also known as wear and tear pigment. The cells of substantia nigra of the midbrain, the cells of substantia nigra in the midbrain is also having the neuromelanin, a pigment known as neuromelanin. These are seen in the substantia nigra of the midbrain. The cells or the soma or cell body is also having the centrioles. In initial or in the ancient time, it was believed that neurons are lacking in centrioles, but after electron microscope, it has clearly proved that neurons are having centrioles which produce and maintain the cytoskeleton of the cells. Centrioles maintain the cytoskeleton. The cytoskeleton of the cell is having microtubules, neurofilaments, and the microfilaments. All three are known as neurofibrils. So a typical cell, the nervous cell or neuron, is having a cytoskeleton formed by the microtubules, neurofilaments, and microfilaments, collectively known as the neurofibrils. The microtubules are having a diameter of 25 nanometers, the neurofilaments having 10 nanometer, and the microfilament is having about 5 nanometer. These are simply the actin filaments and are seen in all contractile cells of the human body and all other animals. The microfilaments are playing a role in axonal transport, the transportation of a substance from the cell body to the terminal part of the axon and from terminal part of the axon to the cell body. So direction from this side to this side is the anterograde exoplasmic transport, and from this to this is retrograde. So transportation from this side to this side is anterograde, and from this side is retrograde. The axonal transport is done by these microtubules. The neurofilaments are maintaining the shape of the nervous cell, and the microfilaments are playing a role in formation of the microtubules and neurofilaments by the centriole. So besides the cell body or soma or perikaryon, the typical neuron is also having some processes, elongated long processes around the cell. In some cases only one process, in some cases it may be two. These cellular or neuronal processes are known as neurites. Neurites. Neurites are of two types. There are multiple small branching, rebranching, and decreasing with the branching. Diameter is decreased with branching. These processes forming a tree-like appearance are also known as arbor or arborization. These small neurites are known as dendrites. Dendrites are receiver. These are receiver, and to receive more and more information in form of action potential, increase their surface area. To receive more information, they divide, redivide, and redivide to form many branches and form a tree-like appearance. These are the dendrites. So here I will explain a single dendrite, the branching pattern like this. again divide, and form all dendrites. These are all dendrites having the microtubules, neurofilaments, microfilaments, and microtubule associated protein also. Dendrites are also having the nasal substance, which is lacking in the single long process, the axon. The axon is not having the nasal substance. And microtubule associated protein, MAP, microtubule associated protein, is also lacking in the axon. So dendrites are forming the arborization or arbor appearance, dividing, again dividing, and diameter is decreasing with the branching of the dendrite. This is known as arborization or arbor pattern. Arbor means tree,